Hello everyone, and welcome back. Hi. Thank you for joining us again. This is our final session for the SPAGN annual meeting, but of course we've saved the best for last. So we now have a research forum for soft tissue sarcoma subtypes, and I'm absolutely honoured and delighted to be joined by 4 international experts on the stage here who will be sharing the latest findings and some promising studies that we hope might change the standard of treatment in the future. So this promises to be a really exciting session. I will kick us off. We're going to begin with a session on leiomyosarcomas presented by Professor Robin Jones to my right here, who is a consultant medical oncologist at the Royal Marsden Hospital in London, and who also is based at the Institute for Cancer Research. So thank you so much, Robin, and I'll hand over. This is your book.
So thank you, Sorrel, for that kind introduction, and thank you for the invitation to speak today. It's really nice to see everybody in person, and just an absolute pleasure to be here. Just quickly start by saying that we were compared to a boy band, but I think we're more like Blondie with Sorrel as Blondie, so that's what I'm going to call her now. Anyway, enough. I've got too many slides, but I'm going to try and keep this focused on really some of the interesting clinical trials that are in development specifically for leiomyosarcomas, and try and put that in context with a number of trials that have been performed over the last few years. So these are my disclosures. I'm going to try and keep this as simple as possible with an initial introduction, again giving the context, and then focusing on localized disease, so leiomyosarcomas that can be removed, and focusing on trials of systemic therapy, either as pre- or post-operative treatment. And then the third component will focus on advanced or metastatic disease when the cancer spread elsewhere in the body, and then finally a conclusion. So I suppose the first thing to really highlight in terms of an introduction and in terms of context is that leiomyosarcoma is one of the more common soft tissue sarcoma subtypes, but this is probably not one disease, and there are many different variants. And you can think of leiomyosarcomas in terms of anatomic subtypes, so there are cutaneous leiomyosarcomas or leiomyosarcomas that affect the skin, leiomyosarcomas that arise in the uterus, in the arms or legs. They arise from blood vessels, they can arise in the abdomen, but also within the bone. So it's clearly a spectrum of different diseases. And I think the other thing to put this in context is that there are different molecular groups of leiomyosarcoma, and Dr. van der Rijn and colleagues have published data supporting the fact that there are different molecular groups of leiomyosarcomas. So this is not one disease. And I hope in the future that we will think of this more in terms of molecular groups, and hopefully incorporate biomarkers into the design of clinical trials. So the second point is the use of chemotherapy, specifically as either pre- or post-operative treatment. I think this in general for soft tissue sarcomas is quite a controversial topic. But over the last few years, I think there's become an increasing acceptance for the use of neoadjuvant chemotherapy in particular subtypes in particular. Leiomyosarcoma is one of those. And this is largely based on a trial performed by Dr. Gronke and colleagues of localized soft tissue sarcomas of the arms, legs, or trunk. And this has focused on patients that have tumours that are high grade, are 5 centimetres or greater, and are deeply seated in the body. And this trial basically had relapse-free survival as the primary endpoint, so the time for the cancer to come back after primary treatment. And the trial essentially allocated people to either have histology-tailored chemotherapy, depending on the particular subtype. So for leiomyosarcoma, this was gemcitabine and dacarbazine, or people were allocated to receive an anthracycline drug, so epirubicin and ifosfamide. And the trial recruited between 2011 and 2016. 435 patients were registered. And basically, this slide gives the breakdown in terms of the types of sarcoma that were recruited into this trial. So myxoid liposarcoma, leiomyosarcoma, undifferentiated pleomorphic sarcoma, synovial sarcoma, and malignant peripheral nerve sheath tumor. And the initial results for this trial showed a benefit, in fact, for the anthracycline plus ifosfamide, both in terms of relapse-free survival, as shown on this curve. So the anthracycline and ifosfamide is the top curve, and this histology-tailored chemotherapy as the bottom curve. And similarly for overall survival, as shown on this curve. And Dr. Gronke and colleagues have published updated results for this trial in 2020 in the Journal of Clinical Oncology, again suggesting a benefit for the use of anthracycline plus ifosfamide in these particular sarcoma subtypes. I think the thing about this trial is that it's in many ways has changed oncologists' opinion regarding the role of preoperative chemotherapy, and a wider acceptance of this plus other data using predictive nomograms has led to a resurgence in the use of neoadjuvant chemotherapy or preoperative chemotherapy for specific histological subtypes, including leiomyosarcoma of the extremity and trunk. In terms of uterine leiomyosarcoma, there was a trial conducted by the Gynecology Oncology Group, randomizing patients after surgery to receive either chemotherapy or no chemotherapy. Unfortunately, this trial failed to recruit for a variety of reasons, and maybe we can discuss this in the question and answer session at the end of my presentation. But there is a current trial in retroperitoneal leiomyosarcomas and liposarcomas run by the EURTC, and this is called STRAS-2. And the design of the trial is shown on this slide. So patients are either randomized to receive surgery or have preoperative chemotherapy followed by surgery. And for leiomyosarcoma, the preoperative chemotherapy is doxorubicin and dacarbazine. So this is a very, very important trial, open in a number of centers in Europe and North America, trying to answer the question regarding the role of chemotherapy in leiomyosarcomas arising in the abdomen. Teeing on to this is a project that we have set up in conjunction with colleagues in Italy and Spain, looking at the use of preoperative chemotherapy in a number of histological subtypes, including leiomyosarcoma, because I suppose one of the major questions is, why do some people benefit from chemotherapy and others do not? And this grant from CRUK basically collects specimens from the preoperative biopsy and the surgical specimen to try and tease out why some people benefit from particular treatments and others do not. This is quite an ambitious project, basically trying to collect data in terms of clinical information, pathology, radiology, but also trying to sequence the tumors, so performing various molecular analysis on the tumor material that's collected, and then also trying to basically look at all of this information in models in the laboratory to see if we can develop ways of predicting response and prognosis, so using artificial intelligence, using nomograms, and also mathematical modeling to try and tease out the differential in response and benefit from systemic therapy in leiomyosarcoma and other sarcomas. So moving on to the third component of this lecture, so advanced and metastatic disease. Of course, in the first-line setting, single-agent doxorubicin remains the standard first-line treatment, despite a number of randomized trials looking at other combinations over the last 10 years. And I think one of the things to highlight is that all of these trials included people with different types of sarcomas. And again, it's very difficult to tease out from these trials, because it's just clinical information, why certain people benefited from treatment, but the overall trial may have been negative. And of course, the most recent of these trials is the PDGFR alpha antibody olaratumab, so a large international phase 3 trial. People were allocated to receive either doxorubicin plus the antibody or doxorubicin plus placebo. And this trial looked at all sarcomas as a group, but also specifically looked at leiomyosarcoma. And I think the notable thing in this trial is that the survival outcomes for patients with advanced leiomyosarcoma have definitely improved over the last 10, 15 years. In terms of the context, there are a number of drugs, of course, that we use in the metastatic setting, and these have been approved by the FDA and other regulatory agencies, so pazopanib, trabectedin, eribulin. Dacarbazine, an older drug, may also have activity in leiomyosarcoma in particular. And moving on to some trials that are currently in development. So the French sarcoma group have performed a randomized trial of doxorubicin plus trabectedin versus doxorubicin based on promising initial data. And they've published and presented these data in over the last two or 3 years. And this trial, as I say, allocated people to either receive the combination of doxorubicin and trabectedin or doxorubicin alone. I'd say that the combination of doxorubicin and trabectedin was more toxic, with more withdrawals. And I think this is a major question in terms of deciding on particular treatment. And again, it comes back to the point that we really need better ways of predicting who's going to benefit from a combination or a specific drug. And in many ways, this is a discussion to have with every single patient in clinic, weighing up the toxicities of individual treatments and the potential benefit. And of course, quality of life evaluation is very important, particularly if somebody goes on to a maintenance treatment, carries on with trabectedin after the combination. And as I say, I think this is a discussion that should be had with every single person in clinic. But as I mentioned, leiomyosarcoma is probably not one disease, and it's probably a group of different molecular entities, and we need better ways of defining which treatment will work for a particular patient. The trial of doxorubicin and trabectedin versus doxorubicin alone met the primary endpoint, or was a positive trial in terms of the progression-free survival, or the length of time for the cancer to get worse. And this is shown on this curve. So for the combination, it was the median progression-free survival was just over 12 months, and for single-agent doxorubicin, 6 months. And similarly for overall survival. But as I say, I think the major question here is the role of combination therapy in weighing up the pros and cons with an individual patient. So this leads nicely into various trials in progress for leiomyosarcoma. So a newer drug, lurbinectedin, which is related to trabectedin and approved for small cell lung cancer, is being considered for investigation in leiomyosarcoma, and PharmaMar are developing a trial evaluating the safety and efficacy of using doxorubicin plus lurbinectedin in leiomyosarcoma as first-line therapy. Another ongoing trial is of a so-called microtubule inhibitor, unesbulin, and the design of this trial is shown on this slide. So people are allocated to either receive the new drug plus dacarbazine or placebo plus dacarbazine. And this trial is currently recruiting in Europe, North America, and around the world. So that, as I mentioned, this is a randomized double-blind placebo-controlled trial. It aims to recruit up to 345 patients, and there are various exploratory endpoints and PK studies, and the randomization is 2-to-1. So out of every 3 people, 2 will be allocated to the combination of unesbulin and dacarbazine, and 1 to placebo plus dacarbazine. And progression-free survival, or the length of time for the cancer to get worse, is the primary endpoint of this trial. And there are various stratification factors, so number of prior lines of therapy, performance status, and anatomic primary site. There's been a lot of interest in PARP inhibitors in leiomyosarcoma in particular. And this ties in with DNA damage response. Working at the Institute of Cancer Research, of course, this is a story that's gone on for decades in terms of the development of this as a therapeutic target, particularly in breast cancer. I won't go into the details of that story, but it's based on this concept of synthetic lethality, where cancer cells with mutated BRCA2 are reliant on another leg of the DNA repair machinery to survive, and the concept is to take out that other leg with a drug. And this figure shows that concept. So on the left-hand side, you can see that if gene A and gene B are working, the cancer cell lives. If one of them isn't working, the cancer cell still lives. But if you block the second gene and the first gene isn't working, then the cancer cell dies. And again, this slide shows that concept, and that's the development of PARP inhibitors is based on this concept of synthetic lethality. So, as I say, this has clearly been an effective approach in various cancers, including breast cancer and ovarian cancer, but there has been interest in sarcomas as well, particularly in leiomyosarcoma, and there are reports of benefit with PARP inhibitors in patients with uterine leiomyosarcoma in particular. And these are some of those data shown on this slide. And this has led to a number of ongoing trials of PARP inhibition in combination with trabectedin in all sarcomas, but a number of leiomyosarcoma-specific trials as well. So an interesting approach, and hopefully a more precise and tailored approach to treatment rather than just treating all of these diseases as one entity. And finally, the EORTC are developing a trial of doxorubicin plus dacarbazine in leiomyosarcomas. So this is based on retrospective data performed by the EORTC of the efficacy of this schedule in leiomyosarcomas, and they're proposing a first-line trial, so comparing doxorubicin plus dacarbazine versus doxorubicin and trabectedin in the context of the French sarcoma group trial. So with that, I'll finish and say that leiomyosarcoma is not one disease. I think in terms of the localized disease setting, there is an increasing awareness of the use of neoadjuvant chemotherapy for sarcomas affecting the arms and legs and trunk. As I said, the STRAS-2 trial is very important in terms of the efficacy of chemotherapy in retroperitoneal or abdominal leiomyosarcomas. And in the advanced disease setting, there are a number of ongoing trials which will give us more information regarding the efficacy of different combinations in this type of sarcoma. But I suppose the major thing to highlight is the importance of doing laboratory work and trying to decipher why some patients benefit from a particular treatment and others don't. And I think in conjunction with colleagues in Italy and Spain, the CR UK grant is very, very important in that respect. So with that, I'll say thank you, and I hope I haven't gone on for too long. I thank all my colleagues at the Institute of Cancer Research and Royal Marsden. Thank you.
Thank you very much, Robin. That was a wonderful talk. So we will move from the world of leiomyosarcoma now onto liposarcoma. And what we will do is we'll take questions at the end. So if you have a question now, write it down, don't forget. We'll take questions after all 4 talks. So I'm really pleased to introduce Professor Peter Reichhardt, who is the Chief Physician in Oncology and Palliative Medicine, and also the head of the Sarcoma Center in Berlin in Germany. So thank you, Peter.
Thank you very much. First of all, I would like to mention my gratitude to be assigned to this boy group, because because as you know, once you reach an age where you would be, in terms of trial inclusion criteria, be put in a study for elderly or frail patients, then you get a little bit concerned. So when we discuss study design and say, well, that's a study especially for the elderly population, I think, oh my God. So, Bernd, I think in a bit more than a decade you will revisit this issue, because he's currently running a study on nearly completing a study in sarcoma looking at elderly patients, and the cutoff is 60. So all of you in the room above 60, that's the poor elderly frail population. Okay. So Robin has already addressed the leiomyosarcoma issue. I will now briefly discuss the liposarcoma. This has, in a way, already been mentioned. Just to remind, sarcoma is a very, very, very big group, and John will discuss the remaining 100 subtypes in the remaining 5 minutes. But just to remind everybody, there's actually 4 major groups in sarcoma family with respect to frequency, and that's most important, and that was very interesting to learn, is GIST, most frequent sarcoma type. Leiomyosarcoma already covered. Next is liposarcoma to be covered now, and the 3rd group is the sarcoma NOS, not otherwise specified, not to be covered today. And all the rest will be addressed by John, and not mentioned on this slide. Desmoids, another story, will be addressed by Bernd. Again, liposarcoma is not one disease. That's a group of diseases, 4 major subgroups. We have the well-differentiated liposarcomas. That's not a benign disease, but not a very aggressive disease. That's by far the biggest group, nearly half of all liposarcomas. Then we have the dedifferentiated lipos, and they always go together. So in a well-diff lipo, there is, in a high percentage, at least a little bit of D-diff lipo, and in a D-diff lipo, there is always some remaining well-diff lipo. So that's actually a mixture. Then we have a different disease, that's the myxoid round cell. So it's the myxoid liposarcomas, and some of them have an amount of round cells. others have not. So that again is two different diseases. So the pure myxoids behave completely different from the myxoid with a high percentage of round cells. The round cells are the bad guys, and the myxoid background is still bad, but not as bad. And then small subgroup, the pleomorphic lipos. So that's again a mixture of different diseases. And we were just chatting in the coffee break. When we give lectures, then we are assigned time slots. And John mentioned he just went to a meeting and there was 1 hour for hormonal receptor positive breast cancer, 1 hour for hormonal receptor negative breast cancer, 1 hour for triple negative, 1 hour for inflammatory, and so on. And there was another half hour for all the sarcomas. So that's the problem, dealing with all these very different subtypes of sarcomas. This just to briefly mention, because I was already ready to ask a question to Robin, but I will ask this later on. What is the appropriate control group in a randomized study? This is retrospective data, looking at what is the best chemotherapy for dedifferentiated liposarcoma. And just briefly, as you can see, the best treatment for these tumors is the combination of doxorubicin and ifosfamide. Robin already mentioned the overall study results is there is no significant benefit for the combo over the single agent. But if you go into more detail and look at the subtypes, it's a different story. So based on this, if you decide on the best possible treatment in a DDIF lipo, again, based on this and our own experience, this would be doxo-ifos if feasible for the patient. Eribulin is another story. There was a randomized study including lyos and liposarcoma. There was a minute difference in progression-free and overall survival, statistically significant but clinically completely meaningless. The difference was actually days, not any meaningful. But when people went into more detail, they found out that there is no difference at all for the leiomyosarcoma, but a significant and, as you can see, clinically meaningful difference for the subgroup of liposarcoma. And therefore, this drug, eribulin, is licensed only for liposarcoma and not any other sarcoma, just lipos. And this difference is meaningful. CDK4/6 inhibition, that's a historical thing to look at. More than 25 years ago, we were discussing using this type of drug in liposarcoma. And there were a lot of discussions going on. We had protocol proposals with the company studying this compound. And the interest of this company dropped overnight. From one day to the other, they stopped thinking about using this drug in sarcoma. Why? It was found out that it's very attractive to study this drug in breast cancer, hormonal receptor positive breast cancer. So they shifted all interest into the breast cancer business and dropped sarcoma overnight. And when you talk to younger people of this specific company, and they addressed me a couple of months ago, we think about CDK4/6 in sarcoma. What do you think? I just laughed in their faces and tell them, young man, no, it was a young lady, if you would be 30 years older, you would know a story. This drug has been studied. There have been seen occasional responses. I would say it's not the #1 study target for liposarcoma, so just mentioning it for the sake of completeness. Another thing is the nuclear export inhibition, selinexor. That was a huge study, global study, studying selinexor in liposarcoma. It was actually the first, and at that time, biggest study looking at this specific subtype. As you can see, there is some activity. When the study was finally done and completed, you see this type of curves, especially the waterfall plot. Everything going down is good, everything going up is bad in this respect, and as you can see, there is not a lot going down, so it's obviously not a very active agent, and there were discussions whether this warrants filing this drug, licensing. In Europe it didn't work, so we don't have this drug available, and based on newer concepts, this actually was not followed any longer. Another interesting, somewhat complicated thing is the idea of using genetically designed agents to address tumor cells. Some tumor cells do carry the so-called NY-ESO1. That's something expressed on the surface, and you can genetically generate T cells, recognizing and hopefully killing these cells. This is actually work in progress. The concept is studied in synovial sarcoma and also in the myxoid liposarcoma. Very small numbers, signs of activity, but up to now, not yet clear whether this will eventually play a role or not. I'm a little bit less enthusiastic about the concept, but we will see, and we're open to see fantastic success if there should be any. Currently, the most interesting issue in D-diff, well-diff liposarcoma is the MDM2 amplification issue. MDM2, I will explain in a minute what this means, is amplified in a number of tumors, in a huge number of tumors, and interestingly, especially for us sarcoma nerds, is that the highest number of MDM2 amplification is seen in sarcoma, namely the well-diff, D-diff liposarcoma, where it's actually 100%, it's part of the definition. So if a pathologist comes up with, oh, this is a D-diff lipo, then it's based on the MDM2 overexpression. So it's part of the definition of the disease. So can we use this all overexpression to do something against the tumors using this as a signal? And yes, we can. So how does it work? There is interaction between P53, something you hear mentioned a lot of times, and MDM2. P53 is a tumor suppressor gene. So something taking care of all the cells, checking that things go well. And if something does not go so well, P53 is supposed to take care of this problem and clear it out. So any type of cellular stress signals, hypoxia, CNA damage, oncogene activation, whatever, this is checked for by P53. They are patrolling everything and checking out whether things go well. And if they detect the problem, they take out the cell with the problem and either repair it or delete it and solve the problem. If P53 works nicely, that's the prerequisite. So we also have MDM2, and MDM2 interacts with P53. Now, we have heard MDM2 overexpression. That's present in D-diff, well-diff liposarcoma. If there is a lot of MDM2, MDM2 is a suppressor of P53. This means if there is a lot of MDM2, this will suppress the P53, and the P53 cannot do what it's supposed to do, control for damage. So if there is damage, it will not be checked, it will not be repaired, it will not be taken out, but it goes directly to oncogenesis. So a lot of MDM2 clears out P53, and the bad signals go through and produce oncogenesis, and the result will be tumor, namely well-diff, D-diff liposarcoma, for example. So what can we do? A possible action would be to stop the overexpressed MDM2. So if we inhibit MDM2, MDM2 cannot inhibit P53. This can restore the activity and check out for the bad signals. That's the concept. Currently, at least 2 agents, MDM2 inhibitors, are in clinical study. And I will briefly, I think I will skip these slides. Just showing you this one. That is a poster for one of the agents. It's called milademetan. This was studied in second-line treatment in comparison to trabectedin. So liposarcoma, pre-treated with doxorubicin-based chemotherapy, the patients were randomized to either milademetan or trabectedin. The study recruited very fast. The results are not yet available, and we will see whether milademetan is better, that's what's expected, than trabectedin in pre-treated patients with D. diff liposarcoma. The second agent is studied in more detail. There are a number of studies going on and data sets. That's a short summary presented half a year ago at the European Society of Medical Oncology meeting by Patrick Szewski. That's the first study in a series of studies, the phase 1 part. We also take part in this phase 1 study, looking at different doses, looking at impact of food and a lot of things, so how does this drug work, what is the optimal dose, and so on. I will skip this, and also this. That's again a waterfall plot, and as you can see, this looks much better because most of it goes down. And again, down is good, up is bad. So majority goes down. That means there is a high efficacy. All the bars are single patients, and a bar going down means in this single patient, the lesion has shrinked, either a little bit or a bit, and the bars going up. In this individual patient, the tumor was growing. And there were more bars going down than going up, so in more patients, the tumor was shrinking than growing. So it's an active drug. That is a so-called swimmer plot. We have fancy names for all these pictures. That's a swimmer plot because all the patients are in their row swimming along, and the longer they go, the better. And the longer these bars are, the longer the patients are on drug. And since you only stay on drug when you first tolerate it, and second, it works, then you are on the drug a long time. You're only for a short time on the drug if you do not tolerate it or if it doesn't work. So if the bar is just 3 weeks, this means after 3 weeks, tumor was already progressing and you're off this drug. So a short bar is very bad and a very long bar is very good. So a lot of long bars, again, signs of activity. That's the D. diff lipose, and that was the well diff lipose. So based on this very interesting results in the phase 1 study, the company decided to bring the drug forward to a randomized study in first-line treatment. That's the Bright Line 1 study. This drug does not yet have a proper name. It's called BI 907828. The BI is for Boehringer Ingelheim, an originally German company, but the home base now is US, as for most of these companies. The study is still in the first part, looking at the optimal dose. The dose is either 45 milligrams or 30 milligrams, still looking at both doses and deciding which is the better one. Better means working better, but still good toleration. So if the higher dose is working a little better, but too toxic, it's not the best dose. So that needs to be finally decided. So that's still going on. That's the so-called phase 2 part, looking at the 2 doses in comparison to single-agent doxorubicin. And that is a small concern. You remember I showed the initial slide. The best treatment for D. diff lipo on the retrospective analysis from the EORTC is doxo plus ifosfamide. Here the control arm is doxorubicin alone. So the question here, and I would like to discuss this later on, are the results, whatever they are, valid? Because the control arm is not necessarily what we, at least myself, consider to be the most active treatment. Nevertheless, this trial will complete the phase 2 part and then move on to the phase 3 part, comparing the optimal dose of this drug to doxorubicin. Just to remind everybody, this drug, either 45 or 30 milligram, is one single pill taken once every 3 weeks. That's interesting. It's not a very complicated schedule. It's not a mouthful of pills. We have studies where you have to use both hands to take all the pills. Here it's a single pill, either a little bit smaller or a little bit bigger, once taken every 3 weeks. That may be the future therapy in first line for D. diff liposarcoma patients. The next generation study with the same drug is using it as part of a combination. That's still in preparation, not running yet, and it will be looking at the same drug, BI 907828, plus enzabenlimab, that's a immunotherapy agent. So we'll combine the MDM2 inhibition with immunotherapy as a combo, and the comparator arm will be one of the standard later line treatments we use in sarcoma, a combination of gemcitabine and docetaxel, and that will be studied in second line, patients having failed anthracycline-based first line therapy. So that's the work in progress. So in summary, currently the most interesting concept in liposarcoma is the MDM2 inhibition. 2 agents under study. One has completed a second line study. One has more or less completed a phase 1 dose evaluation study. Currently, the randomized phase 3 in first line is running fast, and a second line study with a combination including this agent is in preparation and will start probably within the year. Thank you.
Thank you so much, Peter. Right, now we will move to the world of desmoids, and I'm delighted to now welcome Professor Bernd Kasper to the podium, who is a medical oncologist, and he leads the Mannheim Cancer Center in Germany. So thank you, Bernd.
Yeah, sure.
Nice to meet you. Thank you.
Yeah, thank you very much, Sorel, for your kind words, and it's a real pleasure to present or to shift to the desmoids now, and I really have the pleasure to present you what we really call, I would say, a landmark trial, which is definitely a practice-changing trial and will most probably change the whole treatment paradigm for desmoids. So when talking about desmoid tumors, we are talking about a very rare disease. It's a kind of monoclonal fibroblastic proliferation, which is characterized by a very variable presentation and an often unpredictable clinical course, which makes it so difficult for us as physicians to treat. We heard about that TGCT paper this morning. With the desmoids, actually, we were, I would say, the pioneers in this kind of desmoid, in this kind of consensus initiatives. So this desmoid consensus actually dates back almost 10 years to 2014. By that time, we hardly had any evidence for this disease or any level 1 or 2 evidence for this disease. There were no randomized phase 3 trials. There were only very few prospectively conducted studies and meta-analysis. So, and this was actually initiated by SPAGN. We came together in Frankfurt in a rather small group in 2014 that were medical experts from the EORTC soft tissue and bone sarcoma group and a couple of representatives from SPAGN. And what we try to do is we try to harmonize the treatment strategies or recommendations for this disease. By that time focused on sporadic desmoid type fibromatosis. And we published this first position paper in the European Journal of Cancer in 2015 that was then updated in 2017 by a second paper. And 1 year later, in 2018, what we call now the Desmoid Tumor Working Group went global. So we also invited our friends from the US, from Canada, from Asia, and built a bigger group, had an evidence-based literature research for that initiative here, and came up with a global desmoid consensus guideline, which was published early 2020 in the European Journal of Cancer. So this is as a background, so that we already did quite a lot of work for this disease in the last couple of years. And so the treatment paradigm for desmoid tumors is actually active surveillance. So if you have a first diagnosis, the best thing is to do nothing. And so you just have to wait at the very beginning. But when the desmoid is progressively growing, then of course, especially systemic treatment options come into place. And here are the main options that we have at the moment. It's more or less the classic sarcoma-based chemotherapy that can be either doxorubicin or liposomal doxorubicin or other combinations, including methotrexate and vinblastine. Then we very much evaluated the tyrosine kinase inhibitors. We in Germany, for example, did a study with imatinib that was also done in a couple of other countries, in the US, in France. There were altogether 4 studies with imatinib. Then there was, of course, the first phase 3 trial with sorafenib, and pazopanib was evaluated by our French colleagues. But I would like to really focus on the last type of treatment strategies here, and these are the gamma secretase inhibitors, and this is the most recent treatment strategy for desmoid tumors. And I had the pleasure actually to present this trial, the what we called DeFi trial, a phase 3 trial of nirogacestat for progressing desmoid tumors. At the ESMO meeting, Peter already mentioned that meeting that was last year in September in Paris from the European Society of Medical Oncology, and I must really admit that is maybe my personal highlight in my career so far, to present this study in the presidential session. That was a session with, I think, 5,000 or 6,000 people, so this was really impressive. And so, and here is the presentation. These are actually the original slides from the presentation. I think they are quite easy to understand, and it's a very straightforward study. So, the thing is that I talked about desmoids, and the thing is that we tried a couple of different medications, but the problem is we do not have a real approved therapy for that. There is absolutely no health authority approved therapy for desmoids. And so this trial is really, or may change this paradigm, of course. There is a mechanistic rationale for the use of these gamma secretase inhibitors in this disease, as desmoid tumors highly express Notch, and Notch can be blocked by these gamma secretase inhibitors. So this is the rationale behind that, and there were phase 1 and 2 studies with very few patients, altogether 17 patients, and these studies were actually the basis for this phase 3 trial. So the phase 3 trial was then a global, randomized, double-blind, placebo-controlled trial comparing the efficacy and safety tolerability of this drug against placebo. 142 patients were randomized across 37 sites in North America and a couple of countries in Europe. The primary endpoint was progression-free survival, but we also evaluated a couple of secondary endpoints, including objective response. And maybe the most important thing about the trial, a lot of patient-reported outcome measures were evaluated, including symptom burden, physical role functioning, and overall quality of life. So, randomized trial, there were 70 patients in the nirogacestat arm and 72 patients in the placebo arm. It is typical for this disease that we have a very young patient population. The median age was 34 years, and which is also very typical for this disease, that we have a 2/3 female predominance. And then you can see the different tumor localizations. There were quite a high number of multifocal diseases in this study, and you can see the prior therapies. So this is maybe the most important slide. This is the result of the primary endpoint, progression-free survival. We had a 71% reduction in the risk of disease progression in favor of nirogacestat. And the median progression-free survival in the placebo arm was 15.1 months and was not even reached in the treatment arm, in the nirogacestat arm, because we had just there were not enough events. If you look at the overall response rate, which was the first one of the secondary endpoints, we had a 41% objective response in the nirogacestat arm compared to only 8% in the placebo arm, and there were 5 complete responses in the nirogacestat arm where there were no complete responses in the placebo arm. The time to objective response was 5.6 months in the nirogacestat arm and was almost the double in the placebo arm. What is maybe even more striking about this study is not only that we had a reduction in the tumor size. I think what is even more important is that we had an improvement in more or less all the secondary endpoints regarding a lot of different patient-reported outcomes. This is one example. Nirogacestat significantly reduced pain. This was measured by what we call the brief pain inventory short form, which is a kind of questionnaire where you can evaluate the significance of pain. It also significantly reduced the desmoid tumor-specific symptom severity, and it improved the physical role functioning and the overall quality of life. And this was evaluated by a tool which is called GODDESS, which is actually a patient-reported outcome tool that has been specifically developed for desmoid tumors, but we also used a very traditional patient-reported outcome tool, which is called the ERTC QLQ-C30, and we also saw an improvement in the overall quality of life using this tool. Looking at the side effects, the safety profile, here are the main side effects with nirogacestat. That is definitely diarrhea in about 80% of patients, nausea, fatigue, hypophosphatemia, and rash. However, 95% of all these, what we call treatment emergent adverse events, were actually grade 1 or 2, so rather mild. And the first onset of these emergent events, in most cases, in most patients, occurred very early, so just after a few days or weeks during the first cycle. So in summary, this study is definitely so far the largest and maybe most rigorous randomized controlled trial we have for desmoid tumors by that time. And by the way, I mean, gamma secretase inhibitors have initially been developed for Alzheimer's disease, so for a completely different disease. And then a lot of trials were actually underway, but there was never any positive trial with a gamma secretase inhibitor. So this trial is actually the first trial, the first positive trial with a gamma secretase inhibitor in any indication. Nirogacestat demonstrated a very rapid, sustained, and statistically significant improvements in all primary and secondary efficacy endpoints. I already summarized that there was a 71% reduction in the risk of disease progression compared to the placebo arm. The objective response rate was 41% for nirogacestat, including 7% complete responses, and which I think is maybe even more important for the patients, there were meaningful improvements in pain, symptom burden, physical role functioning, and overall quality of life. Nirogacestat showed a manageable safety profile, and 95% of all these adverse events were grade 1 or 2. And it definitely has the potential, I think, to become the standard of care for patients with this disease requiring systemic treatment. And we could actually publish that together with my very nice colleague Mirnal Gounder. We could publish that very prominently in the New England Journal of Medicine just a couple of weeks ago. And let's see, the drug is currently under evaluation by the FDA, and we are of course very curious how that will go out. So they have time until, I think, end of August to come to a decision. We actually expect a decision much earlier. So let's see, maybe in June, when we will have our next consensus meeting at the end of June, maybe by that time we will already have a decision. But this is, by the way, not the only gamma secretase inhibitor which has been evaluated or which is under evaluation. There is a second trial currently ongoing, which is called the Ringside trial, evaluating another gamma secretase inhibitor, which doesn't even have a name yet. It's just called AL102, and it's from a company called Ayala, from a company from Israel, and this compound is currently under evaluation. It is what we call a seamless phase 2/3 study, the phase 2 part of the study, where 3 different dose regimens of this compound is actually already done, and the evaluation came to the result that the 1.2 milligram dose is the best for the phase 3 part, and currently the phase 3 trial is ongoing, comparing this gamma secretase inhibitor against placebo. More or less, this phase 3 part is very much in line with the design of the other trial you saw before. 156 patients will be randomized to that trial. Currently, we have about 60 patients, I think, and this trial is ongoing more or less worldwide. And yeah, let's see, maybe another kid on the block. And yeah, I think that's it. Thank you very much for your attention, and happy to take any questions.
Thank you so much, Bernd. And everybody is much taller than me on this stage. And congratulations again for that incredible work on nirogacestat. It's really exciting to hear the results again. I just want to say thank you. So now we have the rest. So I'm afraid John has the hardest job on this stage, so I want to say thank you for taking on this task. So I'm delighted to introduce Professor John Trent to cover other selected subtypes. So he is the director of the Sarcoma Oncology at the the Director of Sarcoma Oncology at the Sylvester Cancer Center over in Miami in the USA. We're delighted to have him with us today. Thank you, John.
Okay, so good morning. And Sorel told me I had 10 minutes for each of the remaining 172 subtypes. So get ready for the next 28 hours and 40 minutes.
We'll kick you out.
No solos? Okay, so we work with our pathologists closely, and they describe sarcomas and different types of groups that mimic normal structures, and so we kind of think of them this way, although some don't fit into the groups. But there are 175 different subtypes, so everything that we've heard from Peter, Robin, and Bernd are all really pertinent. And so I'm going to talk about a few of the subtypes, but really in the context of what we call precision oncology. And that's really the marriage of understanding the molecular causes of the cancer and identifying medications that can take advantage of those molecular etiologies, much like an Achilles heel. And so sarcoma is unique from many of the carcinomas in that of those 175, a very large number of them have specific mutations. Some of these we've heard about today, like in TGCT. There is a driver mutation that really defines that type of sarcoma. And so that's another way that sarcomas are different, and it's a way that we conceptualize understanding the disease, the biology, and as we'll see, the treatment. Speaking of treatment, we have 175 different sarcomas. Well, we have an increasing number of different therapies. We've heard about some of the chemotherapies today listed here. We've also heard about the targeted therapies here from Bern just now and from Peter. And then we've also have developing fields of immunotherapy in sarcoma, as well as this oncometabolic approach where tumor mutations can result in the production of unique metabolic compounds that don't normally exist in the human body but exist in tumor cells that can also now be targeted to some degree. So in that context, what is precision medicine or precision oncology? It's really, like I mentioned, the underlying molecular etiology that's turning that normal cell into the cancer cell. And that helps our pathologists and us in diagnosis. It also helps us in treatment. And it's important to understand there's different types of NGS that we are doing in clinic now that wouldn't be even possible a few years ago. The first time I was involved in next generation sequencing, where we sequenced the whole exome of a tumor cell, the cost was almost $2 million. And now it's done routinely in cancer patients, and in fact is really critical doing this mutation testing in many of our sarcoma types. We're also developing in sarcoma a field called circulating tumor DNA, where we can actually detect the DNA that is shed from the tumor in a patient's blood to identify these driver mutations, and in some cases identify treatment options for patients. And so I'll discuss a couple of different sarcoma types in the context of precision oncology. And so one type that fits this bill is perivascular epithelioid cell sarcoma. We call it PEComa. It's a rare type of sarcoma that is most often has a mutation in a gene called TSC1 or TSC2, the tuberous sclerosis complex. And so understanding TSC leads to loss of TSC, or inactivation leads to activation of a pathway called mTOR. And there's this new agent that was recently approved in several countries called nab-sirolimus. And nab-sirolimus is an mTOR inhibitor. So TSC is lost in the cell, mTOR is activated, mTOR drives the cancer, and this new agent, nab-sirolimus, can turn off that signal and kill cancer cells. And that was proven in the study called AMPECT, and this is a registration study. Multiple sites around the world were involved where patients received this nab-sirolimus agent, 100 milligrams per meter squared per day, and they were followed over time with the primary endpoint of response rate by RECIST. And so we've seen some waterfall plots today, and we know that when these bar graphs go down, that means the tumor is shrinking. And if you look at the side, on the left side, the y-axis shows the percentage of decrease in tumor size. And so this type of bar graph shows that the majority of patients had some tumor shrinkage when they were treated with nab-sirolimus, particularly those patients with the TSC2 mutation, but also some of the patients with the TSC1 mutation, but not all. And then there were some patients that didn't have TSC1 or TSC2, but still had tumor shrinkage on nab-sirolimus, implying that there may be some pathways that we don't fully understand yet that lead to mTOR activation or are susceptible to mTOR inhibition. And this led to FDA approval. So it was important study for a very rare type of sarcoma, PEComa, much more rare than what you heard from liposarcoma and leiomyosarcoma. Another type of sarcoma is alveolar soft part sarcoma, and this is a really a challenging tumor. Prior to tyrosine kinase inhibitors and immunotherapy, there really was no treatment options for patients with ASPS. Also challenging because these affect younger individuals. So ASPS is an AYA cancer, so it's younger individuals. It also has unusual metastatic pattern with brain involvement, which is uncommon in sarcoma. So it's been a challenge for many years, and then anecdotally, as tyrosine kinase inhibitors came around, the sarcoma community tried them in some patients and some clinical trials. That led to Ian Judson's trial, Cediranib. And again, you can see this waterfall plot showing decrease in tumor size. And then other tyrosine kinase inhibitors were tried in alveolar soft part sarcoma, such as enlotinib, pazopanib, and sunitinib. And what's common about all of these tyrosine kinase inhibitors is that they all target a specific protein, vascular endothelial growth factor receptor. And we think that's the main target in ASPS. We don't know that with 100% certainty, but we do think that that's the main target. Now, interestingly, after TKIs, immunotherapy evolved. And so a number of trials that allowed solid tumors, sarcoma patients. We didn't have a lot of treatment options for alveolar soft part sarcoma patients. So naturally, clinical trial provides good care for patients. And so one such trial was this phase 2 study of tezolizumab ran out of the National Cancer Institute in the US for patients with ASPS. And this study found a 37% response rate, objective response rate. And that number, 37%, tells us that that percentage of patients had 30% decrease in tumor size, or in the final publication submitted to the DNA, 24% of the patients had a 30% or greater decrease in the tumor size that they had. Compared to the alveolar soft part, compared to the tyrosine kinase inhibitors, which also showed some shrinkage, again in the 30 to 35% range. So that really begged the question about what about the combination? We have tyrosine kinase inhibitors that provide some activity and are pretty well tolerated. We have immune checkpoint inhibitors that are active against ASPS that are also pretty well tolerated. But they have modest single agent activity, 30 to 35% tumor shrinkage. They do target VEGF receptor, but that begged the question as to whether or not addition of a VEGF-directed therapy, a VEGF receptor-directed therapy, in combination with an immune checkpoint inhibitor might provide greater efficacy. And the good news is that there were a couple of trials performed around the US that and around the world that included alveolar soft part sarcoma. And I'll show one such trial here, and that's the AXI-Pembro study. The AXI-Pembro study, and in this study, axitinib, a multi-targeted tyrosine kinase inhibitor, was combined with, which targets vascular endothelial growth factor receptor, was combined with an immune checkpoint inhibitor, so an immunotherapy that's designed to remove the camouflage from the tumor cell so that the patient's own immune system can attack the tumor cell. And this combination showed an objective response rate of 60%, meaning 60% of the patients had a 30% or greater decrease in tumor size. And this included a number of patients with alveolar soft part sarcoma, shown on this slide, that are the blue bar. And you see the blue bar is the most common bar that's going down on the waterfall plot and showing the most shrinkage. 60% of patients benefited substantially from this combination. Well, I'll just touch base on a couple of other rare soft tissue sarcomas, and because I think they're important and good examples of precision oncology. One is called dermatofibrosarcoma protuberans, or DFSP, and this is a sarcoma that is caused by a translocation of the collagen promoter getting put in front of the PDGFB ligand, and that causes the normal cells to start producing large amounts of this PDGFB that is a stimulant for proliferation in the normal cell. So the normal cell starts proliferating almost like a desmoid tumor we just heard about. And so these don't metastasize, but they can be locally aggressive. This is a patient of mine that had the DFSP invading the eyelid. The patient saw an ophthalmologist, oncologist, who could only really perform eye removal, an enucleation of the eye. So the patient was referred to us, and I treated the patient with this PDGF receptor inhibitor, in this case imatinib, and that interrupted PDGF stimulating the PGF receptor. So it interrupted the signal just like turning off a light switch, turns off the lights, and the patient had regression of the DFSP from the eyelid back up into the forehead and was able to have an eye-sparing surgery. So we've heard about pexidartinib already and PVNS, TGCT. Another example is denosumab, which is caused by rank ligand, and this interrupts the signaling and is exceptionally active. Again, like 80% of patients benefit from this therapy. The sarcomas such as GIST and fibrosarcoma and some others can have translocation of another protein called TREC, and this is another kinase, and this causes the activation of the receptor and causes the normal tissue cells to proliferate and causes a growth that can be malignant. And luckily, some inhibitors came along, such as larotrectinib and entrectinib. These are approved for sarcomas, so we use them in TREC-fused sarcomas. And then I mentioned these oncometabolites, and so this is really somewhat of a new field that's showing up in not only sarcomas, in this case chondrosarcoma, but in acute leukemias, in brain tumors, and gallbladder tumors. And this whole enzyme pathway leads to production of an enzyme called KG, but in the presence of a mutation in an enzyme called IDH, the KG is converted to an oncometabolite that doesn't normally exist in the body. And that oncometabolite interferes with KG and leads to production of proteins that are normally not in the body, and that leads to tumor cells. Luckily, there are some medicines now that can interrupt this pathway by inhibiting specifically the mutant IDH and not affecting any of the normal cells in the body or normal proteins in the body. So it's exceptionally well tolerated, and about 30% of patients benefit. This is a patient of mine with a conventional chondrosarcoma in the pelvis. Everything on the left panel that you can see that's white is tumor that's contrast enhancing. So that's the chondrosarcoma in the pelvis. I treated the patient for about 4 years on a phase 1 clinical trial with this medicine, ivosidenib, and the patient had remarkable regression of the tumor. And you can see on the right, there's just patchy little areas of continued contrast enhancement. So it can be highly effective in select patients as well. And so that's an overview of some of the principles of precision oncology, some examples, and a few of the 172 remaining sarcomas. Thank you.
Thank you so much, John, and thank you for being very concise with your time as well. You can stay in the band, it's fine. So now we're going to move to questions, so we'll get the mics out and we'll invite some questions from the floor. I think we'll go over there first.
So I'm curious from your medical oncology perspective, so maximal tolerated dose is commonly used as the most feasible option, toxicity equals efficacy often, and so using maybe the GSI as an example, you had 42% have a dose adjustment and 20% discontinue due to GI-related side effects, and we still call it manageable toxicity, right? If you're having so much diarrhea that you're leaving the room, manageable, it depends who you are. So how do you feel that all of you, I'm using that one as an example, fits the paradigm of changing from this idea that maximal tolerated dose is the only way to do these kind of studies? Don't all race.
You know what I think,
but... Well, before I hand over the microphone to my colleagues, you're absolutely right. The concept of maximum tolerated dose is outdated, so to say. That was the concept for looking for activity of classical cytostatic drugs, and even there it doesn't really work. But but most of the drugs we were talking about, or at least some of them, are used on a daily basis or on a weekly basis or on a couple of times per week basis. So you're absolutely right, even a low toxicity, if it's happening over weeks, months, years, that's obviously a different thing. If you get high-dose ifosfamide, that produces a couple of days, not so well-being, but that's a couple of days. And it repeats every 3 weeks, but that's still different to a little bit toxicity, like diarrhea every day and night if it's severe diarrhea. So that's clearly an issue. And many of the concepts presented, especially by John, they don't ask for a highest tolerated dose, because if you are looking for a specific inhibitor, once something it's it's inhibited. If you give more, that doesn't change anything. If you block a lock in the door with something, you can have 100 times more of this blocking thing piling up. It will not change anything. So once a mechanism is shut off, it's shut off. So that's clearly a different story, but you may have seen in the study presented by Bernd, the AL102 agent, actually the one moving on in phase 3 is the lowest dose. So here it was not the maximum tolerated, but it was the dose with expected best balance between activity and tolerability. So in general, you are right, but I think things are moving, and we are looking, or we would like to be looking more into optimal dose instead of highest possible dose.
And that's exactly the point. With the last study I showed was an ivosidenib phase 1 study, and that drug was designed to inhibit mutant IDH. So the rest of the body has none of that. So we didn't keep going to a maximum tolerated dose. We stopped at what we called the maximum biologically required dose when we knew we should be inhibiting 100% of mutant IDH, and it didn't make sense to go any higher.
Yeah, and maybe, I mean, the desmoid example, of course, is a special example, as I'm very much convinced that we don't need the maximum dose here. So, and the thing is, if you have a lower dose, you of course, it's very much better in terms of tolerability and. Let's say, I mean, the most frequent side effect with this is actually diarrhea. And if you just reduce the dose by 2 tablets per day, then this is already much better. And it's quite clear that obviously, if you stop the desmoid from growing at one point, you can go down with the dose, or you can even stop the drug. This is something, of course, this trial is not answering, but it's with tyrosine kinase inhibitor, we usually don't treat the patients for several years. We stop after 9 months or 1 year, and then it's okay.
Just responding to that again, this is a general problem in drug development. I don't want to bash our colleagues from pharmaceutical industry. They have obviously very valid other arguments to follow. But all of us would love to see a number of studies where we look at lower doses, where we look at shorter treatment duration and things like that. As Bernd has just mentioned, all of us are, and most, well, not all, but most of experts are convinced that for desmoid therapy, you don't have a continuous treatment over months or years. We normally stop, and we do see prolonged responses after stopping. So you treat a couple of months, and you follow the patient, and the tumor is shrinking over the months and even over the years. We are all convinced that you don't need to treat them continuously.
How do we
On the other hand,
trials, though, like that? Because, right, that was 150 milligrams was the lowest dose, and that still had 60% requirement for either discontinuation or
yes, so obviously a much lower dose would be sufficient, but it's unlikely that we will ever see that study, and it's unlikely that we will see a study comparing continuous treatment as long as the patient benefits, which may be years, compared to 6-month therapy. Of course, we will not see that study, because for a company that's obviously rather suicidal to do such a study. Nevertheless, it's important, and maybe somebody or we will be able to conduct such a study. But that's clearly a conflict from different perspectives. I mean, if the pharmaceutical industry doesn't earn money, they cannot develop new drugs. So it's, of course, we cannot say you have to give away the drugs for free, but still develop new things. That's not possible. But many of these things, looking at doses, interruption, and so on, are not properly followed based on these conflicting interests.
Yeah, and as I said, I totally agree, and we briefly discussed this earlier on, and I think I like the phrase, it is in a way outdated, and I liked your phrase, Peter, of optimal dose rather than maximum tolerated dose. And just to follow on regarding toxicity as well, if somebody is on an oral medication for a long time, the sort of low, quote-unquote, low-grade toxicity builds up, and it can be really horrible. And I think there's so much scope in a way to prospectively look at optimal treatment schedule, optimal dose, because right now, particularly in gastrointestinal stromal tumor, it's all very sort of, well, we can try this, we can try and split the dose, we can try and take alternate days, etc., etc. But I suppose it's quite expensive to do prospective trials, and that we're sort of almost just trying to tailor it to an individual person based on experience rather than evidence. But a great point, Sydney.
Thank you, guys.
And even beyond that, in the Journal of Clinical Oncology, there is every now and then something called the art of oncology, describing how things should be handled, and that's a very essential issue. With most of these drugs, handling them properly, that's what we can clearly call the art of oncology. And that makes the difference between somebody reading the package insert, looking for a dose, handing it to the patient, never seeing the patient again, and if the patient complains, then referring to somebody else, or really interacting with the patient, because most of these agents, whether being IV or oral, they can be handled very properly according to the specific side effects in a specific patient, and that works very nicely. That concept has basically been developed in the context of TKIs in GIST, but it applies to the same group of drugs in other diseases like renal cancer or whatsoever, but it's always the same concept. So outside a clinical study where things are very strict and rigid, which is of course necessary to develop a drug properly, but outside the context of a study, we have all freedom to handle these drugs as we feel appropriate, and we do have the necessity to increase even beyond standard doses in rare cases. That does exist, but we do lower the doses in a majority of patients below the so-called optimal dose, because in this individual patient a little less works obviously better. But then you have to take into account everything, tolerability, effectivity, and also, and that's the art of oncology.
I think that's a really good point, Peter, and I do like that phrase, the art of oncology. Fitting in with the point regarding toxicity and chronic toxicity, I wrote a piece for the Art of Oncology over 10 years ago, and it was basically based on an interaction with somebody who'd been on a treatment for over 2 years. And I walk into the room and go, oh, this is fantastic. You know, the tumors are still responding. Things are so great. And the person was absolutely, disillusioned with treatment because of these chronic low-grade toxicities, and told me, I hate you, Dr. Jones, this is awful. We reconnected after that, and we carried on with treatment. But it's such a problem in terms of the chronic impact of this on somebody's life, and trying to sort of find that balance in terms of the schedule, in terms of the dose, is in many ways an art rather than an evidence-based approach. So yeah, the art of oncology.
And also toxicity is in the eye of the observer, of course. Some people tolerate some toxicities very well, and others say that's unacceptable. depending on what's your profession, what's your, sorry to say that, intellectual behavior is, whether you are feeling a little bit dizzy, if you are used to reading 8 hours a day, very complicated books, this may be something not be in concordance with your well-being. Somebody never seen a book in the life since ground school will see, well, that doesn't affect me in any way. So these things have to be, again, addressed in a very individual way, and that's what we should take into consideration in these cases, of course.
Yeah, and what we call manageable toxicity
Yes.
is very much different
Blanket.
from the doctor's perspective than from the patient perspective, of course.
And I suppose it may be manageable for a period of time, but the chronic nature builds up.
Thank you, Sydney. You've clearly hit on a very, very hot topic.
Yes.
A final comment from Bernd.
No, I just wanted to give the audience the possibility to have more questions. So.
Me too.
I have a question to Peter about the Bright Line 1 study. You said that the control arm is not considered as the most active treatment. Do you mean that if the control arm had been different, the efficacy of the drug would be less potent as it seems to be today, and that maybe the main interest would be its galenic form?
Well, that's a very important issue, and I would really like to hear the comments from my friends and colleagues. The control arm is normally based on what is thought to be the accepted standard of care, and does not necessarily reflect the individual standard of care of one of the treating experts. Robin has shown a number of randomized studies showing that combo treatment is not statistically superior to single-agent doxorubicin. We have to keep in mind some of these studies had very difficult inclusion criteria, had with respect to the question inside completely improper patient selection. We cannot use a study including all sarcoma subtypes and draw a conclusion that this is a standard of care for this specific subtype. Still, data and guidelines say doxorubicin single agent is a potential standard of care. And of course, companies love to use this as a comparator because it's virtually inactive. The response rate is more or less 7%. Progression-free survival is a couple of months. So if you want to be the winner, you take the weakest possible comparator. On the other hand, and that's my personal criticism, I would never use doxorubicin single agent in a patient with a D-differentiated liposarcoma. I have never treated a patient with a D-diff lipo with doxorubicin single agent because there is no patient, none in the world, that can tolerate full-dose doxo but not doxo plus DTIC. Such type of patient is inexistent. And therefore, I only accept, very difficult for me, doxorubicin single agent in the context of such a study because I want things to develop, I want to see new drugs. And if that's the setting of a clinical study, then we must have to swallow it in a way. But I can only tell right now, if the agent is better than doxorubicin alone, that is only part of the answer. That will not define a new standard from my perspective, because doxorubicin alone is not my standard of care. And being better than something very bad, maybe a little less bad, but still not good enough. So that's a general issue with all the control arms, and it's the same in the study Robin mentioned with the combination with DTIC and the single agent DTIC as control arm. That's also completely inappropriate. We do not participate in the study because we do not see patients in second, third line that have not yet been treated with DTIC. Doxorubicin DTIC is our absolute 100% gold standard in leiomyosarcoma in first line. So a patient in second, third line that has not yet been treated with DTIC does not exist in my institution unless he comes for a third opinion after having been treated outside. So that's an issue. And again, that study, whatever the result is completely meaningless from my position because these patients do not exist, or I should say should not exist.
Yeah, I agree. I think it's a very difficult topic in many ways. I tend to sort of view it in terms of the glass is half full, and that if we do get another drug approved, then that's clearly a good thing, particularly for such a challenging disease as dedifferentiated liposarcoma. But I think we definitely need better ways of teasing out who benefits from a particular treatment. One of the other things regarding dedifferentiated liposarcoma, certainly in our catchment areas, that a lot of people being treated for dedifferentiated liposarcoma have a lot of comorbidities and have a lot of frailty, and sometimes don't tolerate the treatments that are available that well. And it's good to have more novel treatments available that may be better tolerated. But the question of a control arm, I suppose, applies to all of the presentations. So in terms of my presentation, in terms of Peter's presentation, even in terms of desmoid tumor, there are reports of the quote-unquote efficacy of systemic therapy in desmoid tumors going back to the 1970s and the 1980s, and the role of a placebo control arm in a second randomized phase 3 trial, in a way it's a topic for discussion, but also for diseases such as alveolar soft part sarcoma, if you're going to perform a randomized trial, and it's clearly possible to do so. But what is the correct control arm? It's challenging, but I'd view it in terms of trying to get more drugs approved, and the path of least resistance for that.
John, if I may, very briefly, let's assume the study, which I think all of us will assume, BI 90 will be better than doxorubicin alone, may be approved for first-line therapy. Now you're seeing a 23-year-old patient with a large D-diff lipo, potentially but not yet operable. The new standard of care is BI 90. Would you use that or doxoifos to downsize the tumor and enable surgery?
So again, I think the context is really, really important. And in that sort of situation where it's a young patient with very unlikely to have comorbidities, where the aim is to downstage the tumor, I would go for combination chemotherapy because it has more of a track record and can definitely work. But it's important to have the discussion with an individual person regarding the options that are available. And in a way, that was my point regarding the French sarcoma group trial of trabectedin and doxorubicin. That discussion regarding response rate, higher response rate toxicity is something that you need to discuss with everybody. It's not a clear decision, or there's no right or wrong answer. But my preference would be to go for combination chemotherapy for that particular scenario.
John.
Just a couple of comments. One, so I was at an investigator meeting where we're designing a trial with an MDM2 inhibitor and talking about the comparator. And during the background material presentation, there was data out of this institution from their experience that single agent doxorubicin in D-diff lipo has a response rate of 0%. And then in their same institution, doxorubicin plus ifosfamide has an objective response rate of 25%. And so in that meeting, it was very hard for me to understand how they, in view of their own data, were going to recommend single agent doxorubicin as the comparator when they knew from their own experience that doxo-ifos was more active. Unless you want to use a weak comparator to get a drug approved, but that's, bordering on, I think some ethical concerns. And so one of the things that we just submitted for publication is looking at placebos. There's been a lot of sarcoma trials looking at placebos. Other than desmoid tumor or the non-malignant sarcomas at our institution, we don't do placebo trials unless it's a low-grade tumor. For a high-grade aggressive sarcoma, we don't do placebos, and several other centers around the world don't. And so we just submitted a manuscript using some of this data to look at the possibility of developing synthetic control arms. So these are computer-generated Kaplan-Meier curves based on existing data. And sure, they're not going to be perfect, and the technology may not be there, but we're in the era of artificial intelligence, ChatGPT. I may not be here. I may just be some AI bot talking to you right now. So, why can't we start thinking about and moving towards some ways to get away from using placebos or inactive comparator arms in patients with aggressive high-grade malignant sarcomas?
Your point's really well taken, John. I think the question regarding doxorubicin and ifosfamide, though, is very. There are lots of things to take into account, particularly comorbidities, and so doxorubicin and ifosfamide for somebody who has no other medical conditions and is very fit, of course. But if somebody does have impaired kidney function, then doxorubicin is a standard treatment. So, and as I say, in our population, a lot of people unfortunately do have comorbidities. So the trial with a doxorubicin control arm is reasonable.
Absolutely right. Two things, if I may. First, experts like us, sorry to say that, we have a broad armamentarium and we will find the best possible treatment for a patient. If there is a need for treatment, but kidney problems, of course, it will not be doxo-ifos. It might be doxo-DTIC. For other patients, it may be something else, and for the third patient, again, something else, because we can use all that and we make a choice according to the condition, to the goal of treatment, and so on. But the problem and the risk is that in the outside of experienced sites world, people tend to have their preferred treatment, and that size fits all, and they would then treat the patient. I asked you, the 23, I think I said, with inoperable, potentially operable patient, maybe with a pill of BI, because that is the result of this randomized phase 3 study, which may be the result of the study, but still be the completely inappropriate individual choice in this patient. And that is the problem when a study results in a global new standard of care for everybody and everything. That's one issue. And I would like to raise one more point. Or do you want to respond to this?
I just had one addition to that. And so in my practice and in many places around the country, and these aren't around the world, these aren't exact numbers, but for D-diff lipo, for me, the comparator arm that would apply to probably 60% of my patients would be doxo-ifos. There's probably maybe another 30% of my patients that doxodecarbazine would be a good comparator. And then there's probably 10% of patients, these are like over 90 or end-stage renal disease, that I might use single-agent doxorubicin. And so a trial with doxorubicin as a single comparator in a high-grade malignant sarcoma doesn't apply to the vast majority of patients in my study. And if doxorubicin ifosfamide is the comparator arm, then of course I'm not going to put patients on it with comorbidity. That's a different question. How do we manage patients with comorbidity? We shouldn't, I don't think, undertreat patients because of concerns about potential or theoretic comorbidities.
I've often teased John that I should move to Florida because it's so much healthier than inner city London. I do think that there are variations in terms of referral populations, and I think a lot of our patients with dedifferentiated liposarcoma are do have other medical issues, and we have to sort of think more about the selection of combination therapy. I should move to Florida, whoever would.
I mean, the issue is real because a lot of patients have only one kidney.
Well, indeed, and you superimpose other medical conditions, it's tricky.
Again, the risk is.
Let's take the next question. You guys can talk over lunch.
It's really just a comment as the observers in the room watching this dialogue take place on stage. Can you imagine the challenge for patients when we hear your discussion up here and not all of them go to experts like you that take all these factors into consideration? And absolutely, I think, Peter, your point of when it is approved, doctors who aren't like you, that patients may get to, will think this is the treatment that they should give. So this is a fantastic discussion, at all points great, but imagine from the patient perspective, as we sit here and watch all of this happen on stage, we don't always get people like you.
That's a great point, and I think communication is key to all of this, weighing things up with an individual person about the pros and cons and the negatives and positives of a particular treatment approach. I think one of the questions, but I'll go down a rabbit hole now. I better stop, otherwise Sorrel will tell me off. I'll continue with John after the session.
Okay, we have a question over here. Thank you.
I'll open up another rabbit hole, Robin. I echo what's just been said. I think this is a fantastic conversation, and thank you. Now, I've had chemotherapy, cytotoxic chemotherapy. I know what you're talking about when you talk about the side effects. What intrigues me is, albeit 20 years ago, I had single agent ifosfamide in first line because I was selected for that on a clinical study. And you've all mentioned ifosfamide within the context of combinations, but never as a single agent.
So that's what I was actually going to mention to everybody, that very often, particularly with dedifferentiated liposarcoma, our approach, if somebody does have comorbidities, is to use single-agent doxorubicin. I even sometimes use single-agent ifosfamide as first-line therapy and then fall back on doxorubicin. So again, as Peter mentioned, it's trying to use everything, but using treatments in a strategic way. And very often for dedifferentiated liposarcoma, I'd use single-agent ifosfamide, particularly if you can give it as a continuous high-dose schedule, so 14 grams per meter squared over 14 days, 1 gram per meter squared per day. In my opinion, quite well tolerated, and again, taking on board Sydney's point regarding the description and the analysis of toxicity, but it's trying to be thoughtful and trying to be strategic in terms of using treatments. And I suppose, is there a trial doxorubicin plus ifosfamide versus doxorubicin sequentially with ifosfamide? Yeah. Anyway, thanks for going down the rabbit hole, and maybe it wasn't.
We use a lot of single agent ifosfamide. We do it in MPNST, head and neck osteosarcoma, and synovial sarcoma after doxo-ifos. And in the younger, healthier patients, we do high-dose ifosfamide by bolus, 2 grams Q12 times 7 doses, but really that's mostly younger. The other ones we do over 5 or 7 days. And we talked about maximum biologic or optimal therapeutic dose. There are there's data in the literature, older literature albeit, that both doxorubicin and ifosfamide do have a dose-response relationship, so that the higher dose you use, the higher probability you're going to get tumor shrinkage. So maybe that should be revisited, but the older data supports using the higher dose that you think from your experience, and this goes to the art of oncology, and of course discussion with patients about the toxicities, because 2 grams per meter squared of ifosfamide is going to be a lot better tolerated than 14 grams, but it's also going to be less effective. So you have to have that conversation with patients, and shared decision making is what it's all about at the end of the day.
Yeah, mine was 9 grams over 72 hours continuous. But I was intrigued by Robin's point about the 14-day schedule, because 10 years ago when I was diagnosed with a lung metastases, that was one of the options that looked like a viable approach for me, because I'd already responded to ifosfamide earlier. Returning to it would have been logical, but fortunately for me, surgery was feasible, so that never became a real option.
But that's a great point, Roger, because one of the, Jonathan Nugent from Montreal was with us a few years ago and analysed the patients that had been retreated with ifosfamide, and synovial sarcoma and dedifferentiated liposarcoma were 2 types of sarcoma in particular that really benefited from having ifosfamide second, and even in some people 3rd time round. So if options are limited, ifosfamide is a toxic drug, but there are ways of helping with the toxicity and basically helping in terms of getting a response 2nd or 3rd time round.
With respect to high-dose IFOS, John, I think the places are Florida or Berlin. I think we are the #1 consumer of ifosfamide in Germany. I don't think any single institution uses more kilograms of ifosfamide than my institution. So, as John said, it's standard of care in refractory patients, high-dose ifosfamide for many sarcoma subtypes. So that is really an essential role. And some of the old questions like combo or sequential therapy, that's still open. And we have to keep in mind that so-called sequential therapy is in most times a sort of intention to treat. People are thought to start with one and then switch on to the other one, but that doesn't happen. And if we look at the study Robin presented, the doxo-trabectedin study, the concept was doxo plus trabectedin, or doxo actually followed by trabectedin, but followed by took only place in a small minority of patients, which is not our situation. Our next line of therapy, if feasible with trabectedin, is 100% and not 20%. I don't know what the other 80% of patients received. So the idea was give both together or one followed by the other. But as a matter of fact, half of the patient got the combo and the other half got insufficient single agent in whatever in the following lines, but not trabectedin, which is one of the most active drugs in uterine lyo. So again, a big problem in interpretation of the data.
But I suppose that's where, again, communication with the oncologist is so important, you know, to weigh up all these sort of nuanced decision making processes, because very often there's no technically right or wrong answer, and it's a question of weighing up with the individual person what's the right thing for them.
Thank you.
I love the term the art of oncology, Peter. Thank you for that. It is now part of my vocabulary.
You should read the JCO pieces, because
I'm going to talk to you afterwards about this, because on the SPAGN blog, I want an article about the art of oncology. So well, I'll come to you on that, Robin.
But again, I have to admit, it's not my invention. It pre-exists. I would like to claim
I not take
I
the
used
credit,
this phrase
Peter.
years before it appeared in JCO, but I cannot prove that. So,
but I suppose it fits in. I mean, the piece that I wrote, I was so happy because the scan was great, and the person was benefiting for years and years and years. But again, there needs to be the art in there in terms of seeing how that person is feeling and their sort of side effects and the impact on their quality of life. And afterwards, she and I, she just laughed and said, you were so euphoric about this scan, but the distance that I had to travel, the low-grade side effects, it's all needs to be factored into discussions. Anyway, I've said too much.
Okay, I think we
Would you allow a couple more minutes? We started late.
Okay, one final comment.
Difficult. Actually, I wanted to raise another topic to at least address, and I'm specifically looking to Markus Wartenberg, because we have discussed this, and I think it will become much more important in the future, and that's the question of involving patients or patient advocates into the design of clinical studies. And we have heard a couple of examples where randomization process may be difficult because people have pre-existing preferences for either doing this or that, and the more different the approaches are, something like doing everything or doing nothing, that's very, very difficult to randomize.
Can I also ask a question? And I love the concept of the art of oncology, but on the flip side, if the artist isn't you guys, then it's the Wild West of oncology, right? It's what Brandy had mentioned. Well, I'm going to straight call it the Wild West. So how do we take a precision medicine approach but standardize it? So you don't go to Jones and get an entirely different response for another patient as you would, say, Birmingham or a different hospital, where there aren't the artists that are you guys.
Yeah, I think that's a fantastic point. I think, again, it comes down to communication and emphasizing that there's no absolute right or wrong answer and that there are several options. And I always try and tell people, well, this is my sort of preference or my recommendation. Other people may, have a variation or totally disagree about that. But the options are A, B, C, D, and E. And what is the best option for you as an individual person? But I think that there will always be a degree of variation in terms of what doctors, what oncologists think should be the right approach. And I don't think it would be possible to eliminate that completely. I think there are certain things where most people would agree, this is the standard treatment and we should go down this road. But very often there is a lot of there are lots of potential options, and it's important to make it clear to an individual person that there are potential options and to weigh up those negatives and positives with that individual. I hope that sort of answers.
Yeah, I mean, for TGCT, you have risk of recurrence solely based off of academic center. And so you guys are all in a network where you speak to each other because you go to CTOS or ASCO, but there's many providers that don't do that. And so how do we reach those providers collectively at this point?
Yeah,
we try everything we can do to communicate with the oncologists in our area, like Robin said. We present at meetings, we present at regional, I think you said that earlier today too, at regional meetings.
In Hawaii.
And, we try to keep open doors with the patients, the surgeons or internal medicine or medical oncologists in our community. And then the other side of the coin is that we rely on everybody in this room. That's why we're here today. We're here to support you guys so that you can get the word out to the patients. It's not an unusual referral that a patient comes to see us because your group asked them, they came to your group online, asked some questions. Those questions were asked to their doctor, and their doctor didn't know. And they said, well, I know more about TGCT than my orthopedic surgeon, so I don't feel comfortable here. And so they self-referred to one of our places. So So, you know, it's like a lot of things. You have to approach it in as many different ways as you can. I don't think there's going to be one single answer to that difficult question.
Thank you, John. I think that will have to be the final word. Will you join me in thanking our speakers again for an excellent session? And I suppose I should also just remind the band that it's the final of the Eurovision Song Contest tonight. So maybe there could be a late entry. It breaks my heart to tell you that...
I hear 10 points from everybody?
Yes! I can't make them stop now. Okay, well, it breaks my heart to tell you that this is the end of the SPAGN annual conference. So before we close, I want to just say a few thank yous for this wonderful meeting. So most importantly, first up, we should thank the speakers and the chairs of all the sessions today, so I hope you'll join me. I want to give a really, really warm thank you to our organisers, especially Catherine, Mishi, Gabby and Corey. Wonderful. And the technical team that have helped us out enormously and got rid of that horrible buzzing sound yesterday. Thank you. Our wonderful photographer and videographer, thank you for joining us. A big thank you to our sustaining partners for making this meeting possible and allowing us to come together in person and enjoy all of the things that we've enjoyed. It wouldn't be possible without the financial support in order to do this. So thank you, a warmly thank you. I want to say one more congratulations to our Advocacy in Action Award winners. Well done, there were some fantastic projects. And always, I want to thank all of you for wonderful contributions, for everybody being so open in sharing your experiences and creating such a warm, welcoming atmosphere that we've enjoyed throughout this whole meeting. So well done, thank you. And before we go to lunch, as one door closes, another door opens. So if you are joining us for New Horizons Gist after lunch, come back here. If you're staying, that's wonderful, you'll have more discussions. And if you're leaving, we wish you very safe travels onward towards your home. And we hope to see you next year at SPAGN 2024. Thank you. Let's get some lunch.